very complicated process. The first process, which is RNA extraction and synthesis, is a 36-step process, I think. When we first got involved, when they were doing their clinical trials, they actually could not repeat the test. Yeah, there were so many steps and so many places for error that they just were constantly failing the process, even with very skilled operators. Since it was automated, essentially that has eliminated those failure modes, because the machine will do the same thing every single time. So there's actually 3 different platforms that we've put together for them, but the total process end-to-end is fully closed. They are currently in phase 2, phase 3 clinical trials for HIV and renal cell carcinoma. Some of the fascinating things, that's a disposable, believe it or not. But it is still a whole lot cheaper than doing it any other way. So when you look at sort of some of the technologies that are making this possible, this is plastics, it's choosing the right sort of plastics, it's very, very challenging tooling. Yeah, this is a serious piece of engineering in its own right. But once it's developed, you can just churn them out. The KCEP technology, which again is one of these universal technologies, and I think this sort of approach is going to get much wider spread use. And there's certainly been a lot of interest in this core technology. Essentially here, it's a box that's sort of, quite large. It processes about 5 litres per minute of cells. But the key thing here is that it's closed and continuous. The second point about it is that it actually creates a fluidised bed. So what you do is you use centrifugal force to do the separation, but you use a counterflow of fluid to create a fluidised bed. So what you're not doing is compacting cells, which is one of the key problems with any sort of centrifuge. And what we found in this is that we can do very, very fine separation of cell types based on specific gravity. You can separate beads, you can separate cell types. You can actually use this as a perfusion bioreactor. The cells will happily live in there, and you just keep passing the nutrients through the fluidised bed. So this is a rather fascinating technology, and it's a bit of an indication of where some of these processes are going. And I think they're going to change the way that we do some of these cell therapies. We talk about consumables. There's the consumables. So each one of these bags, it's actually a cone shape. It holds a litre. There's 4 of them. There's an umbilical that has 8 tubes. Those tubes are quite large. 8 tubes running through it. It's all fully closed. There are no couplings or anything like that. One of the real challenges here is we're rotating the centrifuge at 1,000 revs per minute. And these plastic tubes and things have to survive that sort of environment. Considerable pressure, temperature, friction, wear on plastics. There's some real challenges in designing these sorts of consumables. And I think if there's an area where, I think it will actually be material science that will be real enablers in this sort of industry. Given how important I think closed processing is as a concept. Now, it's not just Invitec doing this stuff. There are a few other things, and my job is to make sure I know what's out there, because we're putting systems together and we'll use whatever is the right solution from wherever. I can't remember who mentioned cryogenics earlier on today, but cryogenics is in bags. Vials is an open process. So one of the things, and this company here has come up with a lovely little plastic vial that is functionally closed. So now we have something that we can cryo-freeze and we can do the closed process. So these are the sorts of technologies that when we're looking at closing a process, we need to sort of tap into all this stuff that's going on. Most of you will be aware of sort of the way bioreactor kind of techniques. So I'll leave you with that challenge. I think it is always time, but particularly in this area, to look, think and act from a different point of view, because that's what it's going to take.